Hello, my final presentation for Biology 110 will focus on lab number 4, measuring lactase enzymatic activity. In this lab, we will gain a basic knowledge of how lactase enzymatic activity changes over time. First of all, catalyst refers to a substance that increases the rate of a chemical reaction without being consumed. It works by providing an alternative reaction pathway with lower activation energy in order to accelerate the reaction rate. And here, enzymes work as a kind of catalyst. Enzymes have remarkable power to increase reaction rates by at least 1,000,000-fold. Enzymes are usually proteins, although some enzymes are other types of biological molecules. They are highly specific as to substrates and reactions catalyzed. Enzymes function best in dilute aqueous solutions under limited conditions of temperature, pH, and salt concentration. Sometimes, enzymes require one or more non-protein components. They are called coenzymes if they are organic, or cofactors if inorganic. The enzyme of choice in this lab is lactase, which is an example of a commercially available enzyme relevant to human disease practice. It belongs to the beta-galactosidase family of enzymes, and its activity is highly conserved through evolution. We are going to measure enzymes' activity by using spectrophotometry. To achieve this goal, either the substrate or the product of the reaction must absorb light in the UV-Vis spectrum. However, in the hydrolysis of the beta-glycosidic bond in the lactose, none of the molecules do so. To solve this problem, we are going to use another molecule with beta-glycosidic linkage that has similar shape to lactose and can also be hydrolyzed by lactase. It is called OMPG for short. By measuring changes in absorbance, we will get to know how much orthonitrophenol is produced in the reaction in a fixed amount of time. Measurement of the accumulation of orthonitrophenol over time will further indicate the estimation of lactase activity. Equipped with sufficient background information, we come to Procedure 1. First, crush one lactase tablet containing 200 mg of enzyme to an even powder using a mortar and pestle. The powder is then placed on a diagonally folded weighing paper and transferred into a 15 ml tube labeled as suspension. After that, add 10 ml of 100 mm phosphate buffered saline into the tube to suspend the powder, and vortex the tube for 1 minute to maximize enzyme extraction. Label two 1.5 ml centrifuge tubes lactase suspension and transfer 1 ml of suspension into each. This step is followed by centrifuging for 1 minute at 10,000 RPM to sediment solid particles. Transfer 500 microliter of the supernatant from each tube to a clean 1.5 ml tube labeled lactase extract. The optimal temperature for human lactase is 37 degrees Celsius. So, we will be measuring the rate of the reaction at 37 degrees Celsius. Prepare 14 1.5 ml microcentrifuge tubes. Label them separately. There are 2 controls and 12 to be tested. The tested tubes will be separated into 4 groups, with each group has 3 tubes for repetition, as shown in the form. After analyzing the statistical results, we can get the findings that under proper circumstances and within a time period, the longer the reaction takes place, the more product there will be, and the faster the reaction rate is, the more activity the enzymes have. In real life, some individuals have a deficiency or reduced activity of lactase, leading to lactose intolerance. In this situation, body cannot fully digest lactose, resulting in symptoms like bloating, diarrhea, and abdominal discomfort after consuming lactose-containing foods. In the context of human disease practice, lactase can be used to manage lactose intolerance. Lactase supplements are available in the form of tablets or drops. They can be taken before consuming lactose-containing foods. These supplements provide the necessary lactase enzyme to help break down lactose and alleviate symptoms in individuals with lactose intolerance. Lactase is also used in the production of lactose-free or reduced lactose dairy products. These products have been treated with lactase to break down lactose into its simpler components, making them easier to digest for individuals with lactose intolerance. It is worth noticing that lactase supplements and lactose-free products can help manage lactose intolerance, but they do not address the underlying cause of the deficiency. Last but not least, I really appreciate Professor Lee for patient and effortless teaching. Also appreciate all the classmates who offered support. Thank you.